Hello, my name is Susan Pandya. I'm Vice President of Clinical Development at Agios Pharmaceuticals, and I'm here today to discuss the CLARITY study. Thank you for the invitation to present at the Cholangiocarcinoma Foundation's annual conference. It is my great privilege to share with you the progress that we've made with developing ivosidenib in patients with IDH mutated cholangiocarcinoma. These are my disclosures. What is IDH and why did we decide to study it in cholangiocarcinoma? Well, our journey in targeting this molecular marker in clinical trials began in 2014. Idh1 mutations had been discovered in a variety of cancers, including up to 20% of cholangiocarcinomas. Mutations in isocitrate dehydrogenase, otherwise referred to as IDH, lead to the overproduction of an oncometabolite known as 2-HG. High levels of 2-HG have been associated with metabolic dysregulation within the cancer cell, in addition to epigenetic alterations, leaving the cell vulnerable to malignant transformation. Ivosidenib is a first-in-class oral inhibitor of the mutant IDH1 protein, and thus represents a precision medicine approach to targeting this disease. Ivosidenib has been approved in relapse refractory and newly diagnosed acute myelogenous leukemia. The phase 3 CLARITY study was the first pivotal study in an exclusive cholangiocarcinoma population with an IDH mutation. The study design of CLARITY is represented here. As you can see on the left-hand side, eligible patients must have received at least one and no more than 2 prior lines of therapy. Patients must have had a stable performance status of 0 to 1 and measurable disease by RECIST criteria. Patients were randomized in a 2-to-1 fashion to ivosidenib or placebo. At the time of radiographic progression, patients randomized to the placebo arm were given the opportunity to cross over and receive open-label ivosidenib. The primary endpoint of the study was progression-free survival by blinded independent radiology review, and overall survival was a secondary endpoint along with other endpoints listed here. The patient population enrolled are represented in this table. You can see just over half of the patients were treated with 1 prior line of therapy, and over 40% had received 2 prior lines of therapy. The majority of patients had a diagnosis of intrahepatic cholangiocarcinoma, and the majority also had metastatic disease at baseline. The primary endpoint of progression-free survival was met, and these data were previously reported in 2019. Ivosidenib robustly demonstrated an improvement in progression-free survival, as demonstrated by the blue line compared to the red line. The hazard ratio was 0.37, with a one-sided p-value of less than 0.0001. The magnitude of the treatment effect was most pronounced at the 6 and 12 months time points, where a clinically significant proportion of subjects remained progression-free and alive, compared to placebo, where no patients were progression-free or alive at those time points. The treatment effect was consistent across all subgroups. The final survival data were recently presented at ASCO GI this year. Ivosidenib, represented by the blue line, was associated with an improvement in overall survival compared to placebo, represented in the red line. The results, however, were not statistically significant. The hazard ratio for overall survival was 0.79 with a p-value of 0.093. The median overall survival in the ivosidenib arm was 10.3 months compared to 7.5 months in the placebo arm. These data, however, should be considered in the context of 70% of patients who crossed over from placebo to receive ivosidenib. This crossover rate can obscure the true survival effect. To account for the impact of crossover, we applied a well-known method known as the rank-preserving structural failure time, or the RPSFT model. This was a pre-specified analysis. The green curve represents the adjusted survival in the placebo arm. The comparison between this adjustment and the blue ivosidenib arm was significant, with a hazard ratio of 0.49 and a one-sided p-value of less than 0.0001. Importantly, the adjusted overall survival for the placebo arm is very similar to reported survival data published in contemporary randomized trials with an untreated control arm. There were no new safety findings associated with ivosidenib in the study. Frequently reported treatment-emergent adverse events were predominantly low-grade and manageable. Overall, ivosidenib was tolerable with a manageable safety profile that was consistent with the published phase 1 data. Dose modifications and reductions were also infrequent. In summary, the results of the CLARITY study robustly demonstrate the clinical benefit of ivosidenib in patients with advanced, previously treated Idh1 mutant cholangiocarcinoma, as demonstrated by the improvement in progression-free survival and the numeric improvement in overall survival despite a high rate of crossover. The tolerable safety profile and quality of life data also support clinical benefit of ivosidenib. Importantly, the efficacy data supported the inclusion of ivosidenib to a variety of treatment guidelines, including the NCCN last year. We have submitted these data for supplemental new drug application to the FDA and look forward to the potential approval. Now I'd like to pivot and walk you through the timeline for development of the CLARITY study, as I believe this serves as a great example of what can be achieved efficiently in a rare cancer setting when industry, patient advocacy, and the academic community unify around a singular mission of making progress. This timeline depicts the start of our phase 1 program to the recent FDA submission for approval in this rare cancer. We began our interactions with the Cholangiocarcinoma Foundation early in the development of the program. We sought feedback on the trial design at the ICRN meeting in 2016 and incorporated valuable input from the patients and the clinicians who were attending. Our protocol was quickly finalized in late 2016, and we were able to activate the study in the United States before year-end. The trial accrued over a 2-year period of time, and in 2019 and 2020, the data became mature and supported the clinical benefit we hope to see. Now we are awaiting the next steps to finally achieve our goal to have ivosidenib approved and available to patients with Idh mutant cholangiocarcinoma. Operationalizing a study in a rare cancer has many challenges, and the feasibility is often raised as a gate for decision-making. We cast a broad net across several countries to determine the feasibility of enrolling the study. We took a hands-on approach in maintaining momentum on the study as well throughout the enrollment period. Determining which sites to select required detailed questionnaires, multiple contacts, and investigators and research efforts. We were seeking experienced and motivated investigators, and we're very selective in our approach. From the 270 sites contacted, approximately 80 expressed interest, and ultimately we chose 49 sites in 6 countries to participate. One of the benefits of this trial design is that we offered a pre-screening consent, and this helped identify patients early with an Idh1 mutation. This proved to be of great value in identifying patients who might be receiving first or second line chemotherapy and may ultimately choose to then go on to the CLARITY trial. Operationally, this required a very detailed and meticulous effort on the part of several of my colleagues. It also required emphasis on the relationship building with our investigators and the broader patient advocacy community to help instill confidence and credibility in our shared mission. To further raise awareness about the trial, we extended our reach to scientific congresses where we could share and discuss our phase 1 and translational data, advocacy meetings to support the ongoing work of CCF, for example, in making clinical trial information accessible to patients. We also created a digital and print platform to help increase awareness about the disease in the clinical trial. All of these efforts helped spread the word about the study and enabled patient access to critical resources in a timely manner. That concludes my presentation. Once again, I'd like to thank the Cholangiocarcinoma Foundation and the cholangiocarcinoma community, including the patients and families who participated in our studies and helping us make progress in this disease. Thank you.